Should we get started? All right. Okay. Welcome to Wave Club, everybody. My name is Josh Jacobs, and I'm uh, one of the organizers of Wave Club, along with my partners in crime here, Bart Ehrmantraut, Uma Mohan, and Irfan Zabeh, who's right here next to me. It is my pleasure to introduce our speaker for today, who is Elliot Smith from the University of Utah. I happen to know Elliot from a number of years ago, where he used to be here at Columbia with me, and now he is running his own lab at the University of Utah. And he's going to talk about seizure-associated traveling waves in the human brain. One thing that makes Elliot's approach a little different is he is simultaneously, maybe he's going to focus on the epilepsy work today, but in his lab, he's doing both clinical work as well as cognitive neuroscience research as well, which makes his approach in his lab, excuse me, a little distinctive. Anyway, so I'm really looking forward to Elliot's talk. And for anybody who's interested, many of you might be attending the Society for Neuroscience meeting next week. I will be at this meeting, and I happen to know that Elliot will be at this meeting also. So a nice opportunity to get to know Elliot and his work even better would be to go to his posters at the SFN meeting next week. Yeah, thumbs up from Elliot, and to see his wonderful mustache in person as well. So anyway, thank you for coming, and I'm just going to, and I'm really looking forward to Elliot's talk. Just as a reminder, this talk and all of the previous Wave Club talks are archived on our YouTube channel, youtube.com/@WaveClubSeminar, as you can see here. They're easy to find, and thanks to Uma and Irfan for helping put that together for us. And just a couple of the logistics before we get started. The plan for Wave Club is to have about a 40 or 45-minute presentation, and then after that, to make the actual session as interactive as possible. So if you have questions that come to your mind while Elliot is speaking, please type them into the Q&A feature in Zoom. If you have a hard time using the Q&A feature, you can send it to us, but the Q&A feature is the easiest way for us to coordinate the session. And then what we would like to do is to bring anybody who has a question on stage virtually with your camera at the end, and then we can have a conversation with Elliot and all of us together, and making it as interactive as possible. So that's our plan. So anyway, I'm going to turn things over to Elliot. Just as a reminder, every month we have a different speaker. If you're interested in speaking at Wave Club, or you know somebody that you would like to see present, please reach out to me or any of the organizers, and because we're always looking for new speakers for the club. And I'm going to stop my share and turn it over to Elliot, unless I missed anything. Irfan? Okay. Thank you all for coming, and we'll let Elliot take it from here.
Great, thanks so much. Super excited to be here. Yeah, my plan is to start by sharing a bunch of the seizure wave work that I did in my postdoc with Kathy Siobhan at Columbia, where I met Josh and Uma. Then I'll move on just to mention a few of the things that we're kind of going forward with in my lab here in Utah. So seizures have been observed and documented for over 4,000 years. And throughout that time, several salient features have been repeatedly described. Namely that seizures progress slowly through the body, starting in one focal area, especially with motor seizures is visible. And then they exhibit a tonic to clonic transition where people might kind of freeze at first and then start jerking. And then that kind of jerking motion will slow down and then abruptly stop everywhere after it slows. And there's a really great article about the art of epilepsy surgery that documents this kind of long history of people doing epilepsy surgery that I definitely recommend you check out. So we can see these features in this video, this patient who had a kind of facial motor seizure. And I start the video, you can see his certain aspects of his face start to kind of clonically move a little bit and then tighten up. And then that tightening spreads, and then you can see that turn of his face and then the tonic to clonic transition going from this kind of freezing to jerking that will then kind of slow down as the seizure progresses towards termination. And so what hopefully you can notice here is that kind of stereotype progression of the seizure across his face. It should align with kind of the motor homunculus, if you still believe in homunculus. And then as you notice the seizure goes on, you can see the clonic jerking kind of vary across different parts of his face, and then it will slow down as it kind of moves towards termination. So despite this sort of behavior being observed for over 4,000 years, when people started putting electrodes into patients' brains to treat their epilepsy surgically, about little less than 100 years ago, people noticed that seizures spread much quicker in the LFP than would be expected by the semiology or the behavior of the seizure. So this is a kind of an extreme example, but you can see these discharges kind of kick off the seizure here. And then the seizure starts this big bursting activity that indicates the seizure starts spreading extremely quickly throughout the brain, going from the tip of the temporal lobe to the posterior temporal lobe in a matter of seconds. So part of the reason for this kind of disconnect between the recordings in the human brain and the behavioral manifestations of seizures, which weren't like observed in the original animal studies, is this macro-micro disconnect that's very common in epilepsy research. Basically, what this boils down to is that for a long time, human studies were at the centimeter scale in the ECOG LFP or just a scalp LFP, whereas animal studies were induced seizures using some pharmacological agent or an insult most frequently. And then they were done in the level of single units, currents, molecules, cells. So there's this big kind of spatio-temporal disconnect. But from the animal studies, you can see a similar kind of speed of propagation that you would see from the behavioral manifestation of the seizure. And a really good example of that is this study from Andy Trevelyan in 2009, where he recorded from these two patch clamped these two adjacent pyramidal cells in a brain slice, and then induced a seizure with low magnesium solution and 4-aminopyridine. And when he caused a seizure in these brain slices and then got both these cells in voltage clamp, recorded the activity of these brain cells. And since they're in voltage clamp, you can see upward currents are inhibitory and downward currents are excitatory. And you notice that when pyramidal cell 2 had these strong excitatory barrages indicative of a seizure, the pyramidal cell 1 right next to it was getting inhibitory barrages. And that would occur for several seizures in the sequence of this slice, until finally the inhibition, the strong feed-forward inhibition that was impinging on the pyramidal cell 1 was overcome. And you see this kind of tonic bursting followed by this clonic discharging that matched the dominant rhythm of the seizure that both of these cells were then recruited into. And then that kind of overcoming of inhibition into excitation was observed in subsequent seizures in the brain slice. And so then Andy and postdoctoral mentor Kathy termed this the ictal wavefront, which is this failure of the strong feed-forward inhibition among groups of adjacent cells that signaled entry into a seizure or recruitment of a particular cell into a seizure. And so this is in a mouse brain slice, but could we look for this in human recordings. And the strategy there was to use this device that had been developed in the early 2000s for brain-computer interfaces called the Utah Array, which was developed at the University of Utah by Dick Norman, who was on my thesis committee when I did my PhD here. And around the same time, in the mid to early 2000s, there were like three groups of researchers: Kathy, Javon, Sid Cash, and my PhD advisor, Bradley Greger, who were starting to use these in epilepsy patients. And this is a 10 by 10 grid of microelectrodes that penetrate the brain, as seen in previous wave clubs, that allows to record both LFPs from this about the same space as kind of a standard clinical electrode that's used in epilepsy surgery, and isolate individual neurons too. So this has really allowed to researchers to overcome kind of this macro-micro disconnect going from the level of single units to LFPs, and being able to record for weeks in the monitoring unit while patients were having their completely unpredictable seizures, spontaneous seizures. So in doing that, Kathy observed early on a very similar pattern of activity in action potential firing. So these are multi-unit action potentials across different channels on the Utah array. And she saw this slow progression of what they called the ictal wavefront. And that was across the Utah array. And that was followed by this bursting that then kind of locked in with a dominant rhythm of the seizure that as observed on LFP. When you look just outside of the ictal wavefront, you really don't see much change in the action potential firing. If anything, you see kind of a reduction in firing, despite the fact that there might be what appears to be a seizure happening in the LFP. So this was termed the penumbra at the time, the ictal penumbra. And so the ictal just means seizure. Wavefront is kind of this same signature of recruitment that propagates very slowly across the array at about a little less than 1 millimeter per second. And when they looked at the same measure of LFP recruitment, it was about 3 orders of magnitude faster than this neuronal signature of recruitment into the seizure. And so this was kind of what people had seen previously on ECOG, and it really required getting down to the level of individual neurons or multi-units in order to see this signature of seizure recruitment that seemed to match the behavioral manifestations of seizures that have been observed for millennia. So here's a video that's hopefully worth 10,000 words. Basically, what I'll show you here is the closest that Kathy got to recording basically the site of seizure onset. So on the top is the averaged LFP across the Utah array during the beginning of the seizure. And then I'll show you 3 panels. The leftmost panel is the LFP across the microelectrode array. The middle panel is the slow firing rate dynamics. So it's all the multi-unit action potentials across the array smoothed with a 250 millisecond wide Gaussian. And then on the right panel, I'll show you the fast firing rate dynamics. So these are the multi-unit action potentials smoothed with a 10 millisecond Gaussian. And so these should basically show you the dynamics I just described in real time, or 1/8 time. So as the seizure kicks off, you can see these really big discharges happen, and you should see them propagate across the way and just across the array. And just take note of which direction they go. So you should see those kind of propagating towards the left in both the LFP and the unit firing. And then as the seizure kind of builds up, you should start to see some waves start to appear in the early LFP, and you can hopefully keep track of which direction those go. See maybe a few from the left to the right. And then as the ictal wavefront slowly inches across the array, you should start to see some other waves appear kind of on the inside of the seizure focus as it slowly expands across the array. And you can kind of see those coming from the edge of the seizure. So that slowly moving edge of the seizure is the ictal wavefront, and you can see it tends to drive the firing dynamics and the local field potential dynamics of the bursting or the clonic aspect of the seizure as it carries on. And so I'll refer to the inside of this area where that feed-forward inhibition has failed as the recruited part of the seizure. So these are the neurons that are actually producing the seizure, and the LFP is a much larger area of the brain that's being driven by these neurons. Okay, so here is an extreme example of that in time. You can see this, the LFP starting right here on the average trace of the seizure across the array. And this is just the average multi-unit activity here in dark gray. And then this is a raster plot of all the microelectrodes. And you can see as the seizure kicks off here, you get this long period, in this case, like almost a minute of discharging with largely suppressed firing across the array. So probably that feed forward inhibition is in effect here. And then you get this slowly moving wave across the array, which is shown here and down here in the individual channels. And then once this pre-recruitment period is over, you have this post-recruitment period where all the neurons across the microelectrode array lock in with the dominant rhythm of the seizure and start firing following this ictal wavefront. So you can see the slow moving wavefront, and then it's not super clear here, but these are all just kind of like flicking the light switch on and off on all the action potentials across the array. So when I started researching and what I found when I came to Kathy's lab, looking at some of these similar data, is that all of these discharges that happen that make up the seizure, so all these little fluctuations, if we zoom in on them, we notice they were traveling waves that propagated across the array. So these are ranked from blue to white, and you can see a pretty strong spatial gradient across the microelectrode array for both pre-recruitment waves, so these kind of before this little suppressive period, and the post-recruitment waves, which are these kind of after that. So the vast majority of these discharges are traveling waves. And when we looked at the pattern of propagation of these waves relative to the ictal wavefront, which is in the direction shown in white here, we found that the early discharges were going in the same direction as the ictal wavefront. So just like we saw in the video, as the wave moves across, the slow ictal wavefront moves across the array, we have discharges coming out from that. And then once the wave moves through the array, we have discharges going back into the inside of the seizure in the post-recruitment period. And these post-recruitment waves were the strongest effect we saw across our recordings, despite other groups kind of seeing kind of more pre-recruitment waves. So this led us to claim that the ictal wavefront was the source of these discharges, since as the wave moves across, you have discharges coming from the wave this way, and then as it moves the other way, you have discharges coming from the other side. And then the group from Harvard went up like later that year, showed that the wave, like really nicely showed that the waves from the Utah array coupled almost perfectly with the spatial directions of the larger seizure recorded on ECOG. And then Josh Diamond from Karim Zalgul's lab has gone on to do some really nice work looking at similar features of these traveling waves and seizure discharge propagation and ID propagation as well. So as far as traveling wave speeds go, we found that the pre-recruitment discharges were significantly slower than those post-recruitment and those leading up to seizure termination. So we feel like this, or we concluded that this was indicative of this strong feed-forward inhibition being employed early on before the failure of that inhibition led to recruitment into the seizure and then rapid unrestrained propagation of these traveling waves through the brain during the seizure discharges. Okay, so to put this all together, we have kind of three time periods of the seizure. After it starts, there's this pre-recruitment period where the microelectrode array hasn't been recruited into the seizure focus or the ictal core. And so there are these traveling waves, slower traveling waves going out in this direction. As the ictal wavefront moves across the electrode array, you get a change of these traveling wave directions going from the same direction as the pre-recruitment to back inside of the seizure core. And then in the post-recruitment period, you have this reversal of the traveling waves and a significant speed up of the waves going back inside of the seizure. And if you just looked at the ECOG electrodes, you wouldn't be able to necessarily see these firing rate dynamics. And so you would just see coupling of these, a more wide scale coupling of these electrodes, like looking like they're in the seizure, without the knowledge that maybe only one or two of your ECOG electrodes are recruited into the seizure territory itself. And Kathy's done some work kind of like showing how you can pull these different signals apart using clinical electrodes. So hopefully this is clear. I wouldn't expect it to be, but it took me a really long time to kind of understand this. But since we had this really nice empirical finding from our work, we then went on to work with Jun Liu, who's a grad student in Kathy's lab, and Larry Abbott, who he worked with as well, to build a biophysical model of these traveling wave dynamics that we observed across the microelectrode array. And so the way this worked is it's a pretty classic model where we just modeled the change in voltage as a function of several currents, a leak current, excitatory inhibitory currents, and importantly, a slow after hyperpolarization. And then also we had this like recurrently connected field of excitatory neurons with kind of a Gaussian connectivity profile. And another important aspect of the model is that we had an inhibitory feedback onto the like pyramidal neurons that had a slightly wider kernel of connectivity. And so this model reproduced all the spatial features that we saw in the human data. And I'll show you a video of a seizure in the model that hopefully you can appreciate is quite similar to what we observed in actual human Utah array recordings. So as the seizure kicks off, you have this slow expansion of the ictal wavefront, moving from the center of the neural field out to the edges. And it's hard to see, but there's some very small waves going off the edge in this direction, and then much stronger waves going back in towards the inside of the seizure. So it has both the forward propagating discharges and then much stronger backward propagating discharges. And then the model, the ictal wavefront, which was that ring of tonic firing expanding, dies out. The model discharges for a little longer in very kind of chaotic patterns, and then has a few final kind of unidirectional waves before the seizure abruptly terminates. So this really reproduces all those features of the seizures that I mentioned earlier. with some added benefits that the model was able to produce spontaneous seizures after training it to generate a few seizures or inducing a few seizures in it. And then it would also cluster from a focus, which is also similar to human seizures as well. So it would come from the same anatomical area. So here's an example of that. I'll show you the kind of the model space. So each blue dot in this raster plot is a single action potential in this kind of neural sheet. So this little neural space here, and then the x-axis is time. And this is model LFP simulated from the like summed currents across the model. And so when we apply stimulation to the model, you can see it kicks off this ictal wavefront that has these little discharges going out in front of it and much stronger discharges going back inward. And after inducing several of these seizures, the model then learns to this projection bias. It develops this stronger inward connectivity to the source of these seizures wherever we stimulated it before. And that stronger inward projection bias then induces the model to develop its own spontaneous seizures. And so this is a spontaneous seizure from the model. And you'll notice something different here. There are these kind of streaks of firing that occur before the seizure kicks off. And then the seizure itself has kind of a different morphology. It kind of starts with more of a bursting before it goes into the seizure, which we thought were both interesting aspects of the model. But the really interesting aspect is that there are these discharges that occur before the seizure happens. And that's only after you train the model to have a few seizures, it starts to produce these discharges by itself. And these are similar to what are called interictal discharges, meaning they're epileptiform discharges that occur between seizures that are very typical in people with epilepsy. And the super interesting prediction that the model made is that if you look at the direction of these discharges in time, there's a kind of a big burst of firing that's propagating towards the site of eventual seizure onset. And in the simple model, that's probably just clearly because of some added excitability and this inward connectivity bias. But we thought, wow, this is a super fascinating prediction that we could look for in the human data, Utah array data. So people had shown that interictal epileptiform discharges, or IEDs, I'll call them here, propagated. Several papers on them propagating in ECOG electrodes, but no one had really looked on the microelectrode arrays to look for how they related to the seizure onset zone. And there's still a lot left to learn about how interictal discharges relate to the seizure onset zone and how predictive they are of where seizures will start or what they mean for seizure physiology. So these are some classic examples. And what you can see is they basically look like seizure discharges, but they occur maybe every several minutes, and just in the normal background. And there's been some other really cool work showing that they actually can affect cognition quite significantly. So we sought to test the model prediction that these propagate towards the seizure onset zone by first aggregating a bunch of data from Columbia and Utah array data, and then detecting discharges in the weeks of data that we had. So since this is a savvy audience, I'll kind of skip the methods, but we basically detected the discharges. We fit a plane to all of the voltage minima or the maximal descents, and then looked for or classified these as significant traveling waves via permutation testing. Overall, we had 10 patients, 43 days of Utah array recordings. We detected 45,000 IEDs and found that 66% of those were traveling waves. So the majority were traveling waves. And we think that probably more than we really classified were traveling waves, but I think we had a lot of our algorithm was like pretty sensitive. So I think we had a few noise detections in there too. So we then looked at how the pattern of these traveling waves relative to 17 seizures across 7 of the 10 patients that we recorded seizures in. We had seizures in all the patients, but 3 of our microelectrode arrays were never recruited into the ictal core, like I showed you in the video. So we couldn't really look at how like the ictal discharges in those patients. So in looking at these IEDs, we started to notice that, well, that's the best example of what we found. And we showed that the IEDs are going largely in the opposite direction of the direction of seizure expansion, but there's also this kind of bimodality happening. So maybe there's this like overall axis of interictal discharge travel that seemed to kind of align with the direction of the ictal wavefront in this little microspace defined by the Utah array. In looking across, so this looked a lot like what we found in the 2016 paper with the ictal discharges. And in fact, for this patient, when we looked at the interictal discharges in gray, ictal discharges in white, and then the direction of the ictal wavefront, we found this really nice geometrical relationship where the ictal and interictal discharges overlapped in these bimodal patterns, and they kind of aligned on this axis that seemingly was organized by the ictal wavefront. So this was pretty close to what the model predicted, with some notable differences. The first being that when we looked across, this is the best example, when we looked across patients, things are much noisier, as can be expected in biology. But generally, most of the ID distributions, so the gray distributions here, went in the opposite direction, or largely opposite direction of the orange lines, which were the ictal wavefront, indicating that there was some relationship there. And they also seem to align fairly well with the distributions of the ictal discharges. When we looked into these discharges, or when we kind of clustered the directions of these discharges, we found that the majority of the interictal discharge distributions were bimodal, just like we saw with the ictal discharges. So yeah, just kind of like show you the summary stats. Most of the directions were opposed to the ictal wavefront for both seizure discharges and IEDs, and the IEDs tended to line up pretty closely with the seizure discharges, so not super clean. In looking at the bimodality of these distributions, we clustered them into predominant directions and auxiliary directions. The predominant direction being the larger distribution, or the distribution with more discharges in it, and auxiliary being the smaller one. And so we wanted to know if those corresponded in any way with the pre-recruited discharges versus the recruited discharges. So those discharges that are going ahead of the ictal wavefront versus the ones that are going backwards into the seizure. The model would predict that mostly the recruited discharges and the predominant IEDs should have similar features, at least directional features, which they appeared to have. But we wanted to know if there were other aspects of these discharges that corresponded. When we looked just for kind of like physiological differences between the predominant discharges and the auxiliary discharges and the interictals, we couldn't really see much. They had very similar waveforms, very similar firing rates. So we've kind of reasoned that they're probably similar populations. However, when we looked at spatial correlations among the IEDs and the seizure discharges, we found that they tended to correspond across patients quite well. So the directions were similar between the predominant and the recruited discharge subdistributions, and the auxiliary and the pre-recruited subdistributions. And then also the speeds tended to correlate between predominant recruited and then auxiliary and pre-recruited discharges as well. And we only had a subset of patients for this analysis, the ones that were recruited into the seizure onset zone, and then those that exhibited bimodal seizure discharges and IEDs. So bringing this all together, we think this has like important implications for understanding the geometry of human seizures. In seizures, we have this ictal wavefront that's slow moving, kind of moving at the speed of the Jacksonian march that propagates outwards. And you get the smaller distribution of pre-recruited seizure discharges, followed by if you're looking at like a fixed point, followed by an inward going distribution of recruited seizure discharges. And then we found kind of analogous discharges interictally. So maybe that inward projection bias from the model is biasing these interictal discharges to travel in similar directions, in a predominant direction that corresponds with the recruited direction, and then an auxiliary direction that corresponds with the pre-recruited direction. And that these kind of correlate with speed and direction, and then also then the proportion seems to indicate that that could be true. So to bring it all together, the vast majority of ictal discharges are traveling waves, and those extend from the edge of a very slowly expanding wave of tonic firing neurons, the ictal wavefront. And the interictal discharges that appear outside, or the epileptiform discharges often appear outside the area of seizing neurons. So the LFP discharges overestimate the extent of seizing brain. When we built the model of the Utah array data, we found that it predicted these strong inward traveling waves that we then looked for in interictal discharges, which seem to be borne out in our bimodal distributions of traveling waves. And we recently had a paper showing IED traveling waves in SEG that John Rolston was also the senior author on. Okay, so I have one more minute, so I'm going to quickly mention a couple more areas.
Take your time, Elliot. This is interesting.
Okay, all right. So maybe I'll take 5 minutes. But I just wanted to say a couple more, mention a couple more areas that we're super interested in looking into going forward. The first is we realized our amplifiers are direct current, and so we're interested in looking at kind of infraslow activity. And then I'll tell you about some evoked traveling waves and then anesthesia-induced traveling waves. So we've started doing these DC recordings of seizures on our clinical amplifiers. And yeah, when I first started looking at seizures with Kathy, I kind of was like, wow, these are the biggest thing the brain produces. But I have to take that back now because I don't know if you can notice the seizure here, but this DC offset is much larger. It's millivolts relative to kind of the ongoing background activity. So really curious in characterizing the spread of these big traveling waves, potentially traveling waves, or seeing if there is any relationship to the spreading or the spatial features of seizures that we characterize with the Utah array data. We've started looking at some of this, and it does seem that there's some change in like speed of the waves relative to seizure onset. So this is just ranked. This is ranked by seizure onset, so not a ton of organization there. But if you just rank by the spread of the DC wave, you can see kind of like much faster waves going into the onset of the seizure, which is shown in yellow here. And then the DC wave kind of slows down when the seizure happens and then picks up afterwards in some of these channels. So I'm working on this with some other researchers who do animal research or with DC recordings and can actually like image astrocytes, can actually image like, yeah, particular ions that hopefully you can unpack what's going on here with the infraslow activity. And then the second future direction that we're interested in is looking at traveling waves that we're actually evoking with single pulse stimulation. And so Justin Campbell, who's a researcher here, and Corey Inman's lab has been looking at this, where we've been doing a lot of single pulse stimulation on the SEG electrodes. And we're kind of using similar methods that we've used previously to then regress these across space in the brain, in the three-dimensional space of the brain, to look at how they propagate. And then also using methods from Zach Davis and Lyle Mueller to look at this generalized phase measure to see how these waves propagate through the brain as well. And the cool thing here is we can look at just Euclidean distance as well as diffusion image weighted distance, or like DTI, and then also geodesic distance across the surface of the brain, and look at traveling waves for all of those kind of different measures. And it seems like we've found kind of the best fits with the generalized phase measures and geodesic distance, but there's a nice range, and the model fits are actually pretty good overall. And quite a bit of traveling, of like large proportion of traveling waves, though they definitely vary quite a bit across patients and brain areas from where they're evoked. So this would be the percent of traveling waves evoked by stimulation in a particular brain region. So this is online as a preprint right now, is under review. And then the last thing I want to talk about is we just have a few of these Utah recordings as patients went under anesthesia to get their electrodes taken out. And so we're starting to look at traveling waves in this situation, just using really similar methods to the way we measured them with seizure discharges. And you can see this like normal kind of chaotic activity of the brain. This is probably an artifact. And then after anesthesia, you start to get this big discharging or bursting. And then you have this signal that's called burst suppression, where you have these bursts followed by these periods of quiescence. And in looking into these periods, we found that these are accompanied by these kind of strong bursts of multi-unit activity, as well as like delta activity with some kind of bursting in between. And looking at similar methods to the seizure discharges, we found that a lot of these are also traveling waves. In fact, the majority of the bursts are traveling waves as well, in both the LFP and the action potential data as well. So a grad student in our lab, Veronica, is working on that. All right, so not to skim over that too quickly, but I just want to thank everyone from Columbia, namely Kathy and Jin Yu, who really were awesome to work with on this stuff. And then my collaborators in Utah, who led up the IED traveling wave stuff, John Rolston, and Kyle and Bradley, who recorded a lot of the data. And thanks a lot for your attention. Super happy to chat about this more.
Elliot, that was great. Thanks so much. I apologize. There was an issue with the Q&A feature. It seems to be working now. Zoom changed some settings or something, but it seems to be working. So if you have questions, please post them in the Q&A. We're an informal group. We can also do it the old-fashioned way and just talk to Elliot like regular people, as if we were in person, without needing this button-filled device in front of us to type our questions on. I have a bunch of questions. But do you want to go first?
Maybe you can.
I'll go first while we wait for people to type in their questions. One thing that you really piqued my interest on early on in the talk is, I think you made a comment about how the thing on my mind is how much, Elliot, the signals could only be the kinds of patterns you're seeing can only be measured on the Utah rays and could not be measured on larger the macro electrodes used for clinical EEG. Because that feels to me like just an interesting scientific question. It seems like something with real practical implications. Yeah, I don't know. What are your thoughts?
Yeah, definitely. I mean, certainly the Utah array like unpacked a whole new world for understanding seizure physiology, like being able to see the ictal wavefront, that like wave of tonic firing, and then being able to like, as Kathy and Andy did, correlate that speed with the semiology of seizures is just like game changer. Um, yeah, so that was huge. And then like using that, Kathy went on with Shannon Weiss to use the context of the Utah array to really learn a lot more about what signals they could see on ECOG, including like this phase log tie gamma signal, which correlated really well with seizure recruitment. Still hard to see the ictal wavefront really on ECOG, but you could still see that like kind of bursting of high gamma that locked with the seizure discharges, and probably could even infer traveling waves from those, but I don't know if anyone's done that.
Well, that's
Really.
one of the things I'm curious about, like how much you could use. Do we need, okay, like, given that we have some Utah array research like yours and others, is that enough? Do we now know the kinds of signals that we need to know about from these better Utah studies that we can just go and look more use that as background to look more intelligently at the macroelectrode data? Or is there a case to be made that we really should be doing higher resolution recordings in every epilepsy patient to do a better job with them?
Yeah, that's a great question. I think it helps a lot for sure. Definitely provides a lot of context. Um, the there's definitely a balance there, I think,
Why?
with too much resolution of the like too high of resolution of the like large scale uh like brain coverage. It might be just too much information for people to parse for like diagnosis of like and treatment of epilepsy. um I think there are like probably software solutions for that, like clever zooming or aggregation of data or something like that could be really cool. But I know in some of the cases where we've uh, like tried to put in higher density ECOG grids for like research purposes, the neurologists have been a little annoyed with us having to look at all that extra information to
Yeah, but that's
figure out where
software.
the seizures are going.
I mean, my response is, I mean, fine, maybe they're right in the moment, but in
Yeah,
20 years from now, we should have fixed that with, or 1 year from now, we should have fixed it with software, right?
totally. Yeah,
Yeah.
that'd be awesome. Yeah.
There's a bunch of questions from the audience, Josh. So
All
maybe,
right, I'll shut up. I'm sorry. I was excited.
and I have a bunch also, but I might just email you if that's okay, Elliot.
Please, yeah, I'm super happy to take tons of questions offline. So yeah, please email.
Uh, who
This is
wants
great.
to? Yeah, go ahead,
Yeah,
Emma.
we have a couple of really good questions in the Q&A. I see Josh Diamond already has his video up and unmuted. So if you want to go ahead and ask.
Hey, good to see you, Josh.
Yeah, great to see you again, and always good to hear about your work. And um, I sometimes I think about the mechanism of these, is it synaptic or non-synaptic, and or field or gap junctions or what have you. I'd be curious to have you comment on that. But the other angle, and I'd be interested if you thought the same, is sometimes for clinical applications or localizing the source, sometimes it's better to be agnostic about the mechanism because these are such huge phenomena, as you mentioned. One of the biggest things that we can record in the brain, and it's really robust, and that's going to be the case no matter what's causing it, right? So I guess on the one hand, what do you think is the mechanism, or on the other hand, is that something we should be thinking about, or something that would be illuminating to think about?
Yeah, great question. I think like we lean towards synaptic mechanisms because of the speeds and the speed change from pre-recruitment to post-recruitment, I think would suggest that. Um, but that doesn't rule out contribution of other aspects as well. And as far as your second point goes, I don't know. I always think it's important to know like as much as we can, but you're right. I think there's a lot we can do without knowing the exact mechanism as well. But yeah, I would hope that there is something we could learn that would be helpful for developing better treatments. Like, can you stimulate, like if you have a precise RNS, for example, could you stimulate and capture the feed-forward inhibition versus the lack of feed-forward inhibition, and how would that change your ability to disrupt a seizure with like brain stimulation or something like that? Or could you use some sort of active probing technique to understand like network states that where maybe feed-forward inhibition was about to fail or something like that? I think
yeah,
could be
definitely.
super
Yeah,
useful. Yeah.
I see that how like if you're thinking about maybe developing drugs or like you said, neuromodulation, it would certainly help to know what's going on. And I think there's another interesting point which might be underappreciated, but the really macroscopic findings like ECOG and even micro LFP, which is still kind of macroscopic, can tell us about what's going on at the microscopic level, I think, and your work has great evidence of that.
Yeah, yours
I,
as well, for sure. Yeah.
I appreciate it. I would just say one more thing, which is when we're thinking about the wavefront, I often model it as like a focal source, or like in your work, there's this kind of expanding band. But do you think it's strictly a band, or is it more of like a region? Like, do you think the region inside, even the region that's been recruited, is that releasing waves too? Like, is all releasing waves all at once, or is it strictly the kind of front of that region?
Yeah, like our data suggests that it's just a band, and then the waves go inward, basically.
Right.
Okay. But the model suggests that when the wave front dies out, it's like chaotic directions of waves that kind of like have these fractal patterns or something like that, and those persist until the seizure stops. So it's probably really complicated, but some of the seizures definitely look like, especially the ones that have like the early discharging kind of leading into recruitment. It's kind of hard to see the wavefront. It really just looks like the discharges kind of go from more spaced out to getting close enough together that it almost looks like tonic firing, but it might just be like highly overlapping, very rapid discharging, and then that space like spaces out further as the seizure goes on.
Yeah, for sure.
So yeah, not super clear, but yeah, great question.
Thank you.
Awesome, thank you. Li Hao Yan also has a question. I'm gonna ask you to unmute.
Yes. So thanks for the great talk. Since I also work on traveling wave, but I work on the visual stimulated traveling waves. So it's not seizure related. So I want to ask, how should I think about these results for the non-seizure related traveling waves? Have you looked at what's the difference between the traveling waves during seizure and the traveling waves when it's not doing the seizure, basically doing the daily activity of the patients?
Yeah, I mean, there are probably myriad differences. I think just like surface level, like speed and direction sort of differences, but also just like kind of more fundamental mechanistic differences, like whether there is inhibition at play or no inhibition. What's driving these traveling waves? Is it some process that's propagating across a gradient or a cortical map or something like that? Or is it like an interictal discharge? Yeah, probably a ton of differences. Yeah.
I have a question, if that's okay, Josh.
Of course, Bart. Dive in.
Yeah, two questions. You keep talking about feed forward inhibition. Isn't this inhibition that's coming from the excitatory isn't it more feedback inhibition from the neighboring neurons that are excited? That's one question. And then you guys don't take into account like I've seen a lot of models for seizure propagation that are related to ionic accumulation of like potassium propagating across, and that gives it's sort of related to spreading depression and things like that. I don't know what the speed of migraines and spreading depression is, but it seemed like it was on the order of millimeters per second. So I was curious if there was any connection there.
Yeah, I think that's super interesting question that like there's a lot more research to be done on related to the spreading depression. It's similar. I think it's a little slower than seizure propagation from what I've seen from like the calcium videos. And there's this guy, Matt Stern at Emory, has just published a couple papers on this, looking at calcium dynamics, calcium imaging of seizure-associated spreading depression. It's really nice work. So that would be definitely something to check out. As far as, yeah, and I think like that super related to like glial cell, like shuttling of ions and stuff like that. Like basically how can these like inherent inhibitory mechanisms, like are they overcome just at the like more support level rather than like interneuron level? I think could probably be really important, but it's like totally unknown as well. I think that's going to be a really active area of research going forward.
So you really think, I actually think the slow propagation of the seizure is purely, I mean, you got it in your model. I have to take a look more carefully at the model. So it's purely just synaptic recruitment and loss of balance of EI, I guess. Is that?
Yeah, I think so. I mean, that's what the model would suggest, but obviously we didn't include glial cells or ion dynamics in there. So it's kind of like, it's definitely a blind spot for sure. I think it'd be really cool to look at that. And I saw a talk by Ivan Soltesz a few years ago where he showed like this like step change in glial cell. It was like the ability of glial cells to like shuttle ions around, and that was like highly associated with like future seizure states. And now people are starting to see these kind of step changes when they do like active probing stimulation. Like there's a paper from Graham et al. In Brain last year that could be somewhat related to that. So I think it's like a huge like dark horse in the field that could be really cool to look into. Yeah. Mm-hmm. But yeah, our model, it's a big blind spot in our model and research for sure. Yeah. And then yeah, I think they always just call it feed-forward inhibition because it's basically reactive inhibition to the like strong feed forward signals from the seizure. Yeah, that's your first question. I don't know if that's a useful term, right?
That's pretty wild. Thank you. We have one more question from the chat. Harsh, if you want to go ahead and ask.
Yeah. Thanks. This is super interesting. When the traveling waves are propagating inward from the ictal wavefront, is there a point of convergence? Is that point of convergence the initial start of the ictal wavefront? Are they related? Is it just going into the center, and if the shape is nice and spherical, then it goes to the centroid, and if it's not nice and spherical, then it goes wherever? Like I don't know if you guys have looked at that, but that at least, and I know the graphic you showed was of a model, but it in that model it didn't go to the center. It actually was off center in its convergence.
Yeah, exactly. And it didn't really go, play that again. It didn't really go to the center in any of the models.
Interesting.
Yeah, so something about the dynamics of the model where it kind of tended to come from somewhere. Maybe at the beginning it's kind of going to the center. But in truth, we just didn't have the spatial spread to record the center of the seizure in any of the human data. So we can't really give you an empirical answer for that, only a theoretical answer. And in the model, the actual wave directions are kind of highly susceptible to like very small changes. They're kind of chaotic. Yeah.
And are patients getting, where in each patient is the Utah array implanted?
So it varies for everyone. The goal is to get it as close as possible to the seizure focus or the site of seizure onset for each individual patient. Most of them in Utah were in the middle temporal gyrus, like anterior middle temporal gyrus. At Columbia, they were kind of more spread out, like kind of all over the lateral temporal lobe, frontal, several in the kind of middle frontal gyrus and 1 or 2 in the parietal lobe, I think. Yeah, so kind of all over the place.
Got it. Thank you.
But the goal is to get as close as possible to the side of seizure onset. And I think 9 out of 10 of the rays that Kathy implanted were resected eventually, like right in the onset zone.
Got it. Cool. Thank you.
That was really great, Elliot. This is super thought-provoking. Thank you so much. I have a lot of thoughts now about how we can use how maybe we should be using high-resolution electrodes for epilepsy and cognitive stuff and all of these things. I've been familiar with the papers, but I haven't seen the movies like this, and it was very cool. Very neat.
Yeah, thanks so much.
Yeah, so thank you for coming and sharing your work with us. And
yeah, my pleasure.
Yeah. All right, so we're going to wrap up now. Thank you everyone for coming. Thanks for all the organizers' work at UMA. The recording will be posted online if you want to share it with your friends or review or whatever. Take care. Maybe I'll see some of you at SFN. And thanks for coming to Wave Club. Thanks again, Elliot. This was great.
Thanks so much.